Okay, the last session was about quality control, self-filtering, gene filtering. That's what you successfully managed to do. And now let us talk about normalization. This morning I showed you this plot about transcriptome and genome mapping. So we have the information of a transcript. We have a short piece of these transcripts, so that's what's depicted here in purple and orange. And these pieces are now mapped to the genome. What's particular about these short pieces is that usually introns are already excluded from the transcript. Not always, but most of the time. That's why we have to make some kind of a gapped mapping. So that's shown here with these dotted V shapes that's connecting two parts. That's about the mapper. You don't have to worry about these things. The current mappers tend to handle this pretty well. So let me now point out what's the difference between what you have to think of when you have different protocols. Here we have mainly problems with a difference with full-length protocols and free prime protocols. So with the full-length protocols, you get reads from the full transcript. So from the full length of the transcript, you have these kind of pieces. In a free prime protocol, you only have the last part of the transcript. So everything that's adjacent to the polyadenylated sequence, so the poly A tail. That means that in the full-length protocol, the gene length affects the number of captured molecules. If you have a short gene, you get fewer reads from it compared to a long gene. When you only have reads from the free prime end of your transcript, this gene length does not affect the number of captured molecules. So no matter what kind of gene you have, they always give about the same number of molecules. So that's why in a full-length protocol you should consider the gene length in the normalization, and that's usually done in the so-called TPM normalization, so transcript per million per gene length. That's the abbreviation that we have here. In contrast, when you have free prime bias, so the data that you have at hand, every molecule contributes equally in the normalization, so you go with so-called CPM normalization. That's counts per million, so that means you divide by the total number of counts that you have per cell. Or you can use a single cell specific approach, which is a so-called SCRAN, that takes also into account cell type specific differences. So how can they look like? I told you in the morning about this so-called V shape that you can have when you look at the number of genes versus the number of counts. So library size versus number of genes that are expressed. So that means that some cells might have a lot of reads, so huge library size, but express a few different genes. So they're really specific to a certain genetic program. And those should be handled differently than cells that have a lot of genes and also a lot of counts. So, and this is what SCRAN does. So it clusters the data first and then computes a size factor that is cell type specific. So to encircle this again, we see here these are size factors that are specific for one particular population, and here we have the other part of our V shape. So cell type specific size factors lead to a more accurate normalization of the data and help to reduce cell type specific biases in the data. If you don't have this kind of V shape, then it's also okay to go with the CPM normalization. So basically dividing by the number of counts that you have. That's not much of an issue here. Okay, so again, cell type specific characteristics are reflected with Scrum. Unfortunately, Scrum is an R package, so that's one of the reasons why we have to use RPy2 to connect our Python notebooks with R. Okay, that's normalization in a nutshell. One last point on this. I showed you 3 types of normalizations. I think there's around 30 around that you can encounter. Yeah, so these 3 types are like the most common ones that you can use. Okay, my favorite topic, batch effect correction. I think that's a love-hate relationship, at least on my side. I would say like 90% of my PhD went into that topic. So I try to cut that story as short as possible. So meet the chicken. The chicken is a data set from the gut epithelium and consists of 14 different samples. What you see here is a low-dimensional representation, so 2D plot, and every cell is colored by the batch. And what you see is this really nice banded structure here, here on the chest of my chicken. And also here this huge tail that's also consisting of different cell blocks. When you encounter this kind of banded structure, that's a batch effect for sure, because the cells that belong to one batch cluster as one. So the batch effect is the dominant factor in the data, and that is a strong bias that you would like to get rid of. So after batch effect correction, data should rather look like this, so that the batches are nicely overlapping, and at least from the visual inspection point of view, you can see that the data is nicely overlapping. Again, there's at least 20 different methods to tackle batch effects in data, and most of them try to map the data from different batches, here colored, so different colors, and try to match them. However, you also have the biological component. Imagine you have different cell types here depicted as different shapes, and you would like to have the different cell types to cluster together instead of having all of the cells in one big blob. So this situation, when you see all of your different cell types overlap, that's certainly a sign of overcorrection. Okay, we just conducted a huge benchmarking study to see which method helps us best to correct for batch effects while preserving the biological signal. I show you a plot later on this. The long story short, it depends. And what we also have to consider when it comes to batch effect correction is that the different methods create different outputs. So what does it mean, output? We have 3 different ways. Either we have a corrected feature matrix. That means that our counts or our normalized counts are batch effect corrected and then returned as a corrected matrix. Or we have an embedding. So that means a 2D representation of our data, just like the one we have here on the left-hand side. So some methods just give you an embedding, and then you have to continue to work on this embedding while keeping the raw counts. Or even more general, you can have a so-called graph object. That means that you only integrate the cells in a way that you change the neighborhood of cells. So then this method only returns the information this cell is close to these other cells. That's even less information. You can still create an embedding from it, but certainly it's super difficult to create a corrected gene output from it. So you don't get these kind of things. So when you would like to look at corrected gene data, you cannot go with those two. Okay. So that's the take-home message from our benchmarking study conducted by my colleague Malte as leading author. And what you see on the left-hand side, that's a plot that Luke actually created. So talking about label conservation versus spectral effect correction. So what you see here is how batch effect correction, so that's scored from 0 to 1, should perform best. So best methods are at about 1, and how they preserve the biological signal. So that would be also like 1 would be best and 0 would be worse. So most of the methods are rather in this top right score regime, which is quite good. So this is where they should belong. But what we also see when we check where we have the unintegrated data, oh, no, we don't have it. Anyways, so what you see is that none of the methods really go to the top here. So they do some kind of correction, but they also tend to remove some level of the biological signal. So that's always entangled. On the right-hand side, you see our top scoring, and we tested over several parameters, not parameters, but different scenarios, from small data sets to really complex data sets with huge samples and many samples, many cell types, to a data set that has almost 1 million cells. And you see here the overall ranking. So in our test cases, we saw that BBKNN did a really good job, but it's a graph method, so it's a bit trickier to handle the rest of the things. Second one is Scanorama, and yeah, you can read the ranking yourself. Some methods do not scale well, so like Seurat and the Trevi. This is where you have no results here because they needed too much computational resources to be computed on 100,000 cells or 1 million cells, respectively. Last point, what we can take home from this huge study is that selecting highly variable genes first is a good thing to do, so it improves the performance of batch effect correction in general. So how do we select highly variable genes? Here's one plot that is standard output from scanpy. And you see here the mean expression of every gene versus the dispersion. So dispersion is considered as some kind of measure for variance that is normalized by the mean. Okay, when we don't normalize the dispersion, we see this kind of decrease from with higher mean expression. So genes that are lowly expressed tend to have a higher variance or a higher dispersion. When we select highly variable genes, we have to take this into account, either by normalizing the dispersion and then make bins. So we bin the genes, or we sort the genes by the mean expression and then group them in several bins, and then decide for each bin which of the genes is highly variable or not. So that's the so-called Seurat approach to select highly variable genes. In contrast, you can also go with the cell ranger way to select highly variable genes, and there you can also fix a number of highly variable genes that you would like to have returned. So that means if I want to look at the 2000 top highly variable genes, I would go with the Sura flavor of selecting these kind of genes. Also in the tutorial we go with the Sura way to select highly variable genes. I won't say that one way is better than the other. I would like to point out that the Cell Ranger flavor worked better in my my studies, because sometimes the Sura flavor kicked out the marker gene that I would really loved to have in my data. But that's some point where you can decide which one works better for you. Okay. Let's have a brief look at embeddings and visualizations of your data. There's different ways. For example, a so-called PCA. PCA is a linear embedding method that basically rotates your data along the strongest variance. So you compute the variance of your data, and then you make a rotation of the data such that PC1 shows you most of the variance and PC2 shows you the second most variance of your data. So that's a linear embedding. Often PCA is also used as a pre-processing step to reduce the noise in the data. A second one is the TSNE plot, so stochastic neighborhood embedding. That's a stochastic method that optimizes neighborhoods. So you compute the cell neighborhood in a high-dimensional space and try to conserve this neighborhood relation in the low-dimensional space. It uses the t-distribution to accomplish this task, and that usually leads to a very clustered structure of the data. Diffusion maps, in contrast, can be considered as some kind of non-linear version of PCA. Also, because it reduces the intrinsic noise along a so-called trajectory. So diffusion maps tend to pronounce trajectories, and if you have a disconnected object, so we have cells that are really far apart, diffusion map is not a really good way to show them, because then this difference between two really disconnected groups of cells is pronounced, and the heterogeneity within these two groups is not resolved anymore. Okay, when you look at UMaps, they tend to combine the advantages of a t-SNE and the diffusion map, to be honest. So you can compare the UMap plot from its concept to a t-SNE plot. The advantage of a UMap is that you can compute it in a much faster. So when a t-SNE needs minutes, a UMap needs seconds. Okay, the second thing is that UMap pronounces transition much better, so the global structure of the data is resolved in a much better way. From the concept, they're really similar. So also UMap tries to compute a neighborhood and embeds this in the low-dimensional space. Finally, I also would like to show you a so-called force atlas and the star shape, and what you use here is some kind of molecular dynamics approach to embed data. So cells that are similar to each other try to stick together, and you have a repulsive force for cells that are more dissimilar, so they're pushed away. And in this way, the embedding tries to embed cells in 2D. Here is a so-called scree plot. That's connected to the PCA, and we rank the PCA by the variance contribution it has. So we have more or less two parts here. So one is the strongly decreasing slope, also strongly decreasing variance in the first few components, and then we have a really, really long tail of slowly decreasing variance contribution. This long tail is considered as picking up the noise in the PCA. So these components are not really informative for the analysis. So you would like to focus on the first few components and also for computation of the low-dimensional embeddings. And what you here now see with the elbow is a so-called elbow method. So I would like to distinguish between noisy principal components and informative principal components. And I try to find the elbow of this plot. So that's the long tail, that's the steep slope, and somewhere here is the point where the elbow is. So that's why it's called elbow method. All right, I already spent a lot of time on this, but never mind. Let's go for the third session and tackle all these kind of issues. So we go for normalization, we go for batch effect correction, at least in the more complex setting, and we go for visualization of the data. If your sample has, or if your data set has only one sample, don't worry. You can read up on batch effect correction as well, or visit the more complex tutorial.